Okay. Hello everyone, my name is Mackenna. I'm a junior, and over this past year I've been conducting research on the detection of a presence of antibiotic-resistant pathogens in Oahu's waters. Thank you all for coming. Okay, so first off, you may be thinking, what is an antibiotic-resistant pathogen? Well, antibiotic-resistant pathogens are types of infectious bacteria that have gained resistance to the antibiotics used to treat them. A common infection here in Hawaii that is caused by an antibiotic-resistant pathogen is MRSA, or methicillin-resistant Staphylococcus aureus. Antibiotic resistance occurs naturally, but over these past decades, antibiotic resistance has been exacerbated by the misuse or overuse of antibiotics. That is because environments with both high counts of bacteria and antibiotics have perfect conditions to create antibiotic-resistant bacteria, the two most common breeding grounds being hospitals and wastewater. These are suitable environments for this process because in large counts of bacteria, there will be a majority of susceptible bacteria and a small minority of antibiotic-resistant bacteria. The large amounts of antibiotics will kill off the majority of bacteria, and so the minority of antibiotic-resistant bacteria is left to become the majority. This population can grow larger without competition and even transfer their resistant genes to other bacteria. One factor that makes these environments more common in Hawaii is cesspool leakage. Cesspools are these underground human fecal wastewater storage tanks that don't treat the wastewater but just concentrate it in one location. This often causes wastewater to leak into surrounding water resources and contaminates the waters with high counts of bacteria and antibiotics, creating the previously mentioned perfect environment for the antibiotic-resistant processes to take place. According to the DOH, there are currently 88,000 cesspools in Hawaii, and nearly half of them are leaking, releasing 53 million gallons of untreated wastewater into the ground each day, which in effect increases the likelihood of antibiotic-resistant pathogens being created and then infecting people in that community. This is a lot of information being thrown at you guys, so why should you care? Well, the increase of antibiotic resistance among different bacteria is a global threat to human health because of the dangers of infection. Antibiotic resistance decreases the effectiveness of antibiotics used to treat infections, which increases hospitalization length, medical costs, and mortality, with an approximated sum of 700,000 deaths each year. Between now and the year 2050, the World Health Organization predicts that antibiotic resistance will cause 10 million deaths, an economic crisis as severe as the 2008 financial crisis, and will force 24 million people into severe poverty. In other words, antibiotic resistance is dangerous, and it will affect all of us in different ways. For some, it may be even detrimental. Personally, that's why I took interest in this topic, and that's why I decided to conduct research on it in specific areas of our community in order to help create more knowledge on these dangers and raise awareness of them too. So what areas am I talking about? I chose to sample the Kahaluu area just north of Kaneohe and the Alawai Canal, which runs right past our school, because of their history of cesspool leakage, high bacteria counts, and infection reports. I also chose these areas because local residents continued to recreate in them despite state warnings. So these guys on the left are outrigger paddlers, but I also know that our paddling team here at Iolani School paddles in the Alawai Canal, and this guy on the right is a fisherman from the Kahaluu area, and on my trips out there, I've seen people like him, fishermen, or boaters, paddlers, and even a boy playing with his dogs in the stream, despite the many signs posted that say keep out of water, just like this one. However, you can't blame these residents because it has been years since these signs have been posted and this issue is still prevailing, mainly because of the lack of awareness on this topic. So in order to understand this issue more and create more data on it, I had two goals. My first goal was to sample the test sites for the levels of Enterococcus, for the levels of Enterococcus and E. coli as an indication of fecal contamination and water quality. I did this so that I could predetermine the likeliness of finding a presence of antibiotic-resistant bacteria and possibly antibiotic-resistant pathogens as a result of fecal contamination. I hypothesized that if there were high levels of Enterococcus and E. coli, then the area likely experiences cesspool leakage and contamination, so therefore there would be a presence of antibiotic-resistant pathogens in the site. First off, I use IDEX call alert kits to quantify the levels of Enterococcus and E. coli in 100 milliliters of each water sample. I analyze these kits by counting the number of yellow wells, which produce a most probable number of Enterococcus colony-forming units per 100 milliliters of water. I did the same for E. coli, but I counted the number of fluorescent wells instead. From these levels, I found that high levels of Enterococcus and E. coli existed in the Kahalu'u area, and high levels of Enterococcus and moderate levels of E. coli in the Alawai Canal existed. In the Kahalu'u area, Enterococcus levels ranged from 12,247 colony-forming units to 31,859 colony-forming units. And in the Alawai Canal, average Enterococcus levels range from 833 colony-forming units to 4,611 colony-forming units. For Hawaii, the statistical threshold value for which should not be exceeded for safety reasons is 130 colony-forming units, and all of these levels have widely surpassed this safety limit. In the Kahalu'u area for E. coli levels, they range from 670 colony-forming units to 1,622 colony-forming units. In the Alawai Canal, E. coli levels range from 31 colony-forming units to 109. For Hawaii, the statistical threshold value for E. coli is 410 colony-forming units, which the Kahalu'u area surpassed, but the Alawai stayed under it. But this is so concerning because it passes the geometric mean level, which is what the tendency of the E. coli levels should be in the water. So after confirming high levels of Enterococcus and E. coli, I moved on to my second goal, which was to identify pathogens from the test site and test them for resistance against ciprofloxacin, gentamicin, vancomycin, penicillin, erythromycin, and tetracycline, in order to find a presence of antibiotic-resistant pathogens. I chose these 6 antibiotics because they are all used in different ways to treat bacterial infections. One way is by attacking the cell walls of the bacteria, attacking nucleic acids that produce DNA, attacking nucleic acids that produce RNA, or attacking the proteins that produce ribosomes. So to start testing pathogens for antibiotic resistance, I had to identify them first. To do that, I grew bacteria from the water samples on agar. Then I identified them using 16S sequencing, which requires 3 parts to the procedure. First, I had to PCR the bacteria to amplify the DNA, then use gel electrophoresis to visualize them, and then send off my samples to be sequenced so that I could identify them online. By identifying the species, I could identify whether or not the samples were pathogenic. And so from the Kahalu'u bacteria samples, 54.2% of the samples were pathogenic, 37 were not pathogenic, and 8.3% of the samples were not determined. This was pretty similar to the Alawai Canal bacteria, which 66.7% of were pathogenic, 25% were not, and 8.3% were not determined. And after I identified the pathogens out of the bacteria, I then tested the pathogens against antibiotics using the Kirby-Bauer method. To start this method, I inoculated a pathogenic colony in Kirby-Bauer broth and grew it up to a 0.5 McFarland standard turbidity. I then plated this onto an agar plate, stamped it with the 6 antibiotics, and let the bacteria grow overnight. The next day, I measured the diameter around the antibiotic stamp to determine resistance. If the diameter was not present or was smaller than the zone of inhibition, then the bacteria grew despite having the antibiotic stamped onto it, and so it can be considered resistant. But if the diameter around the antibiotic was larger than the zone of inhibition, then the antibiotic killed the growing bacteria and the bacteria is susceptible to it. And from the Kirby-Bauer method, a presence of resistant pathogens to all 6 antibiotics was found in both locations except for ciprofloxacin resistance in the Kahalu'u area. However, finding ciprofloxacin resistance in the Alawai Canal is very concerning because cipro plays an important role in the treatment of severe infections, as they are one of the few available treatments for severe Salmonella and E. coli. So here are some of the pathogens that I found and their antibiotic resistance types. From the Kahaluu area, there were 3 pathogens which were susceptible to all 6 antibiotics. Psychobacter species, which can cause endocarditis or peritonitis, was ciprofloxacin resistant. 4 pathogens were resistant to multiple drugs, one of them being Pseudomonas species, which can cause UTIs, pneumonia, endocarditis, and other infections. And this was resistant to penicillin, vancomycin, and gentamicin. From the Alawai Canal, only 1 pathogen was susceptible to all 6 antibiotics, and the rest were resistant to multiple. 2 of these bacteria were Vibrio species and Vibrio huangiensis, which causes cholera. And Vibrio species was resistant to gentamicin and erythromycin, and Vibrio huangiensis was resistant to penicillin, vancomycin, and erythromycin. Another bacteria was Aeromonas hydrophylla, which causes septicemia, meningitis, and endocarditis. And this was resistant to tetracycline, penicillin, vancomycin, and erythromycin. So, in summary, high levels of bacteria such as Enterococcus and E. coli and a presence of pathogenic resistance to all 6 antibiotics tested suggest that these waters experience wastewater contamination, and it supports my hypothesis that these areas are highly unsafe for recreation. And as always, scientific research projects have errors and difficulties. Some of the difficulties that I ran into during my project were dealing with the naturally occurring Enterococci in Hawaii's waters and weather events such as heavy rainfall. These factors affected the Enterococcus levels that I measured. I also had to deal with some limitations of the Kirby-Bauer method. Because most of my bacteria samples were from brackish or marine bacteria and the Kirby-Bauer method is tailored towards freshwater bacteria, I had to use a different agar medium to grow the bacteria on to allow for survival. And some further projects or implications of my research include running another series of tests on a site with unpolluted water to serve as a control comparison. Or I could compile my data as a report on the health dangers of bacterial contamination in recreational waters for submission to the DOH. I also could share my findings with listeners to increase public awareness of these health dangers. Lastly, I want to offer some advice for future research students. The most important piece of advice that I can give is to pick a topic that you're passionate about. It's easy for anyone to get burned out or uninterested in a topic after doing research on it for nearly a whole year. But if you pick a topic that you're interested in, there's a chance that you're going to stay motivated throughout your research process. This perseverance is also something that you will need, because there are going to be so many difficulties that you'll face as you develop your methods, perform them, then write your paper and present your findings. But you shouldn't give up, because you'll learn so many new things throughout this process that it will all be worth it. Lastly, I want to give a big thank you to all my mentors, Dr. Dulai and Dr. Chan, my parents, my brother Matthew, and my amazing friends for supporting me through this journey. These are my references, and thank you for coming.